All right, in this video we're going to be reviewing Unit 3 Cellular Energetics for AP Biology. My name is Mr. Chipman and I teach AP Biology at Murray High School in Murray, Kentucky. In this video we're going to be looking at photosynthesis in particular, going through all the facets of that. Photosynthesis is about energy storage. What do we mean by that? Well, the process of photosynthesis creates glucose. Glucose is something that organisms need in order to use energy. There are certain kinds of organisms that make that those are usually like plants or autotrophs. Autotrophs are the bigger group. Plants, of course, are a type of autotroph. And in order to do photosynthesis, we need the sun. Photo, light, synthesis, make things. So make things using the sun, those things being glucose and oxygen as a byproduct. What do we need in order to do that? Well, we need carbon dioxide and water. Sometimes this process is called carbon fixation. Called fixation because think about it, when you fix something, what are you doing? You're taking something that's unusable and making it usable. Carbon dioxide is unusable by most living things, and so they take that unusable thing, make it into a usable component, which all living things can use. Glucose is universal in its usage and being able to use for energy. Water is also necessary for this process. You get glucose and oxygen as byproduct. Here is the overview of photosynthesis, in that photosynthesis is composed of two reactions. You have your light reactions on the left and the Calvin cycle on the right. Sometimes you'll call it the light, this is sometimes called the light dependent reaction, and on the right you'll hear this called the light independent reaction as well. AP literature almost exclusively uses the term Calvin cycle, but light independent makes sense because you don't need light to do this, whereas light dependent, you do need light in order to accomplish this reaction. Let's go a little bit further. Don't know why that penguin's there. This is also how oxygen was originally put into the atmosphere. Originally aerobic organisms, or anaerobic organisms, that did photosynthesis, you produced oxygen as a byproduct. Oxygen filled the atmosphere, also allowed for the creation of ozone, which blocked UV light, allowed organisms to move closer to the surface, start to breathe oxygen, through that process of natural selection. And the first aerobic organisms came into being. Let's look at those two pathways again, be a little more specific this time. You have your light-dependent reactions, and this picture is great, by the way. This picture, I tell my students that this is kind of their pathway to understanding things. As far as, if you got this picture down, you can really just tell me all about photosynthesis. All of the nitpicky details aren't all that important if you don't understand this big picture. Light-dependent reaction, of course it needs light. Light comes from the sun. This is happening on the thylakoid membrane as well. That's what this is a picture of, the thylakoid. This whole thing is a chloroplast. Water is necessary in order to do this. The purpose of water is that water is split to provide a steady stream of electrons into this process. Those electrons are going to be picked up by this molecule here. Well, actually it's this one up here called NADP+. NADP+ is an electron carrier. It's going to pick up those electrons, turn into NADPH, and it's headed to the Calvin cycle. Well, ATP is also made in the light reaction, also headed to the Calvin cycle, both of them to expend their energy, which is why they don't have the little boom signals around them. After they leave the Calvin cycle, going to be filled up, and so this is a coupled reaction here, and these two reactions are also coupled in their products and reactants. Well, this whole thing is coupled with cellular respiration in its products and reactions. And so let's get a little more in depth. Let's look at the light reactions. For the light-dependent reaction, of course, light is necessary. Light is going to be absorbed by these structures called photosystems. Sorry, just checking my thing real quick. Okay, good. Photosystems. Photosystems 2 and photosystems 1. Here they're in reverse order for whatever reason. And the reason that they're in reverse order is because I believe they were discovered that way. Photosystem 1 was discovered first, photosystem 2 discovered second, but then they discovered that photosystem 2 actually comes before photosystem 1. All that is completely unimportant. Light is being absorbed by both photosystems. These are just big proteins that have a lot of pigments in them. Pigments are light-absorbing molecules. And so on photosystem 2, water is split to do a couple of things, actually. Oxygen is going to be let off in this portion of the reaction, and this is going to cause oxygen to be a byproduct of this reaction. Protons are also going to be produced in this reaction. Protons are going to be built up inside the thylakoid as a way of kind of building a concentration gradient that we'll use later. And then those electrons are going to travel through this little system right here that is called the electron transport chain, using a series of redox reactions or redux. There's oxidation reduction reactions. Not a whole lot on that in the curriculum now, but oxidation reduction is basically the idea that they're just passing these electrons. As a protein gains 2 electrons, its charge is going to be reduced, and as it gets rid of those electrons, that process is called oxidation. Very simple idea. You don't need to know the names of these proteins, not important. And they're going through this, as they go through that, what that's doing is it's powering the passage of these protons that are being made into this thylakoid lumen, which is going to kind of build up, almost blowing up like a balloon. Think of it that way. It's not really that, but it's building up a gradient, right? Those electrons are going to be, they're going to travel to photosystem 1 and eventually going to move on down to this protein here. Its name is unimportant, and that is where NADP plus is going to pick up those electrons and then carry them off to the Calvin cycle. These electrons have the sun's energy in them. That is important. We're going to talk about where those electrons originate as well, and then that concentration gradient is going to be used by this molecule here, ATP synthase, one of the most important proteins in the body or in any living system, because this protein is basically going to facilitate diffusion across this membrane, high concentration in the lumen to a low concentration outside the lumen, and that concentration gradient is going to be used to create ATP. That ATP headed to the Calvin cycle to do ATP things. What's going on in the Calvin cycle? Well, there's a lot going on. We're not going to go into all the details here. It's not all that important. AP has stripped this down almost completely, which is okay for us. We're going to just talk about a couple of the main ideas. So the CO2 is the big driver for the Calvin cycle as far as, let me just make sure. Okay. CO2 is the big driver for the Calvin cycle as far as this is going to be how the main reactant, where the carbon's going to come from. CO2 is unusable by most organisms. In fact, it's dangerous to a lot of organisms. But glucose is not. And so the Calvin cycle is where those unusable CO2s are going to be created and made into glucose. Glucose has energy in it. Where does it get that energy? Well, it gets from the ATP and NADPH that are made in the light-dependent reaction. So here comes ATP, here comes NADPH, and there's 6 of them. Why are there 6 of them? Well, because there's 6 carbons in the glucose. Each one of those carbons is going to kind of be energized. You can think of it that way. There's a whole lot more going on here. Remember we talked about the pathway reactions in the enzyme video? Well, this is a giant pathway. Every one of these steps has some sort of enzyme associated with it. Lots of names. None of that's important. The only enzyme that you might see is this enzyme here, ribisco, thought to be the most abundant chemical in the world, or most abundant organic molecule in the world, ribisco, because just of all the plant life. And ribisco is going to take those carbons, put them on the merry-go-round, and then when they spin off, they're going to turn into G3P. 2 of these G3P make a glucose, and G3P is used for the plant or by organisms like plants for lots of different things, but most of the time it's going to be combined with another G3P to make a 6-carbon molecule called glucose. Glucose is where we're going to get all of our energy from. All these other things, not all that important in this class, thankfully. Let's talk about chlorophyll. Chlorophyll is the primary pigment that is found in plants. Chlorophyll is significant because it has magnesium in it. Well, what's magnesium? What's so significant about magnesium? Well, it has 2 valence electrons, and it's those 2 electrons that are going to power this whole reaction. If we go back up here, should be a 2e, really, because there's 2 electrons that are going through this whole process. And those 2 electrons are going to be supplied by magnesium initially. Well, magnesium only has 2 valence electrons, and so water is going to keep that supply going. And that's why that magnesium is significant in the middle of chlorophyll, because it's basically just constantly getting rid of those, adding them and getting rid of them, like a non-stop transfer kind of thing. So chlorophyll is very significant. That's what this is showing you too. Sometimes you'll see this picture. All these cytochrome complexes and all that kind of stuff, not important to this class. Understanding the main idea, really and truly, if you can just get this picture down, understanding that light is being used here in order to break down water to make oxygen as a byproduct, in order that you can have ATP and NADPH to feed into the Calvin cycle to energize those carbons that are coming from the air in order to make glucose, you're going to be pretty good. All this other stuff is really too much. Photosystems, just to mention those again, these are large pigments that are gathering light. There's a lot of things going on here with funneling light and different kinds of pigments found. There's a whole lot more here than we need to know. Right? You may see that on the exam, but that you can't let that scare you, because as long as you get the big idea, all those details should know by now anyway, that the exam is going to spell a lot of those details out for you. It's just going to leave the main ideas, the basics for you to bring into that. And so, the difference between chlorophyll A and B and all that stuff, it's just trivia, all right? And the exam will give you the trivia you need to know, but the basic idea is what you need to be able to bring to the test. And so make sure that you know the basic ideas, and I feel like this video is going to be helpful for you in that regard. If it was, please please comment below, tell me that was good. Also subscribe to this channel. I'll be producing lots more videos in the days to come and in the years to come. So if you recommend me to someone else, keep thinking about me when you get to college and you're like, I don't get this stuff because my professor isn't actually a good teacher, then you can just refer back to my videos, because I enjoy doing this. Again, like and subscribe. Thank you for watching. Good luck on the exam.